and millions of different and specific antigen binding receptors for the B and the T lymphocytes out of like 30,000 human genes. This is really the trick that evolution has solved, and which is rather surprising. And there was a long debate before the molecular biology was invented whether this goes like using just a single gene or a few genes generating mutations in them, and then or else we have as many genes as many VCRs. And the final answer is that we have a relatively large set of gene segments that are designed and code protein sequences that are involved in the antigen binding by the T and B cell receptors. For the antibody, we discussed this. If you take myeloma patients and isolate the light chain of the myeloma-secreted monoclonal antibodies from the urine of these patients. So, myeloma patients have a monoclonal plasmacytoma, which means that each of the cancerous cells will secrete one type of light chain and one type of heavy chain. The light chain is at so high concentration that it is going to show up in the urine. In the 60s or something, even late 50s, they realized that. They isolated using biochemical methods the light chain from the urine, and they used classical organic chemistry methods to sequence these proteins. Each individual has a different B cell plasmacytoma. Each B cell would be secreting a single and presumably different antibody. And this is what they found. They found that there is a region, a large region of the light chain called the constant region, which will have no mutations when you compare the protein sequence. The same amino acids all over, few mutations, like this little text. When they looked at the N-terminal part of the molecule, they got plenty of mutations. Look at this. There's a lot of mutations, and all the places where you see letters, you will have a different amino acid at that position among these proteins. So they concluded that only one part of the antibody, presumably same for the heavy chain, is variable, and the other one is very conserved. So what they suggested is that only the V segments need to be different, so maybe one or a few constant region encoding gene segments are enough to encode the constant region of the light chain or the constant regions of the heavy chain, and only the variable gene segments would be different. Come on in. And this was the hypothesis, and this is entirely true. So how this vast diversity is generated could be so that you only have a constant gene segment, a single one, and after that there's a gene rearrangement process that takes place at the lymphocyte development. I will be speaking even louder if you keep talking in the back, because it's irritating. I'm not angry, I'm just raising my voice. And I want you to stop talking to each other, okay? Is that clear? Thank you. Okay, thank you so much. But really, and I will bring a microphone next time if Gabor let me come again, because I want to have fun here, not get angry. It's Friday night, okay? It's not good to piss me off before I go home and beat up my children, okay? So that's really not the point. So constant region and many variable regions. So the process which actually takes place and is an integral part of B cell development is to choose one V segment and fuse it with the constant segment. As it turns out, the V segment is assembled by multiple further gene segments. So look, these are like exons. These red ones encode the V segments. The yellow ones contain a joining segment, and the constant region encoding gene segments, only four for the lambda chain and a single one for the kappa chain, codes for the constant region. For the heavy chain, there's approximately 40 different red ones encoding the heavy chain those segments. There is 6 encoding the J segments, and then the D segments, which are missing from here in the light chain, are approximately 23. Now, when the V cell develops, first the heavy chain rearranges, and one randomly chosen V segment is fused, one randomly chosen J segment is fused with a D segment, and then after that, a second rearrangement occurs when the V segments are randomly joined with the D segments. So these guys are important. Now, see what happens. The easiest thing is to look at the kappa chain. In this case, the V kappa 2 gene segment is chosen with the J kappa 5 segment. But you could generate possibly 5 times 35 different combinations of this, so it's quite high number. And this is put together as a single segment encoding the kappa chain, again during development.
So only the kappas are getting together? Or
yes.
It could be kappa or alpha?
Again?
It could be the kappa or alpha together, or just only the kappa and alpha?
The V regions of the kappa will join with the J regions of the kappa. It's only the kappa. The lambda chain is somewhere else, and the heavy chain, see the lambda is on chromosome 22, and this one is chromosome 2. So this is within the same chromosome. Now,
Go on.
So, now, it's okay that we put together these gene segments, but why these gene segments are not expressed before they rearrange? That's interesting because they are there. Each one of them has a leader sequence, a promoter, and so on, but they somehow doesn't get expressed before they actually got rearranged. That would be a mess to actually express the 200 different variations at the same time. So before the rearrangement, there's no expression, and the key thing is why they are not expressed, because they need an enhancer sequence to help them to get expressed. And before the rearrangement process, like this, when one of the V chain is joined with the joining segment, the promoter is too far from this enhancer associated with the kappa chain constant region. As soon as they become in close proximity, same for the heavy chain, the promoter gets activated and transcribes the primary RNA transcript, which is then spliced, as you learned in biochemistry, and the mature mRNA can get translated and results in protein. Okay, so in the developing immune-globulin genes, structural changes occur. This was a great surprise because at the time, everybody thought that one gene, one protein, and that the genes are steady and they don't mess with each other, except that there's like crossovers, but only when the gametes are generated. Now this has not been true for anymore after this. So the rearrangement process takes place in every single developing B cell. But those enzymes that do this rearrangement process, they are not expressed in any other cell type, just in the lymphocytes. Okay, so they are called RAC enzymes. This requires a single recombination event. Now what about the heavy chain locus? The heavy chain locus has this extra green D segment. So there's more combinations to be generated for the heavy chain, because there's like, how many? It was 23 D segments in the human genome for the heavy chain. And for the constant region, you have 4 domains of the mu chain. The mu is the first to rearrange. 4 exons encode these 4 domains. So in heavy chain, you got 2 rearrangement process, one in between the D and the J segment, and one in between the DJ and the V segment. The guy who produced these figures, I believe, was the person who discovered the I-10 motifs. A very famous person.
What is the G stand for?
The J?
D.
Diversity regions. That's how they called it. They are just very similar, these gene segments. So, two rearrangement for the heavy chain and a single rearrangement for the light chain. Now I want you to think about how many different combinations could this possibly generate. By the way, when you generate a heavy chain and the light chain, they can randomly form tetramer BCRs together. Theoretically, there are no restraints of randomly combining these chains. This is an animated slide, so when this heavy chain is generated, the variable region is highlighted. This can combine with this light chain or any others. Okay, light chain, same, different heavy chain, different light chain, and so on. Combinations are many, depending on which gene segment is picked. Now, how many can we generate altogether? This is shown here, the light chain, the kappa light chain, one isotype of the light chain, has 35 or so variable segments. The lambda is around 30 as well. The heavy chain may have 46, depending on who has it. I mean, that's usually different in various different people. Joining segments are there in each one of them, 5, 4, or 6. These segments are only present in the heavy chain, and the constant region has not too many, but it doesn't really add anything to the binding specificity of the receptor. As the combination and the rearrangement is random, you can generate heavy chains 46 times 23. Where the hell is 23? Sorry, here. 23 times 6, because the V, D, and the J segments can randomly recombine. That makes it 6,000 different heavy chains. You could generate 38 times 5, which is here, kappa chains, and lambda chains you would have 33 times 5 at maximum. So that makes it 355. Altogether, 190 plus 165, 366, 55, and any heavy chain can recombine with any light chain to form a viable BCR. That means 6,300 something sort times 355, 2 million different copies based on the number of gene segments. Is 2 million receptor, or would 2 million receptor be enough to fight all sorts of bacteria, viruses, and so on? No. So the whole procedure is designed to be lousy. Moreover, there are mechanisms that are that will generate diversity at the junction point. As the short segments, the 2 million different combinations, would be put together, before they join, there are enzymes that add extra nucleotide in a random fashion, chosen out of the 4 different nucleotides. Therefore, you will add new sequences in a random fashion that can generate, instead of 2 million different receptors, you could have 30 million times more, okay? So, I stated and we calculated that 2 times 10 to the 6 receptor can be generated out of this many gene segments. And this was the so-called combinatorial diversity. And now you have another type of diversity which is generated by extra nucleotides that were not even part of the germline, not even part of the genome. This type of diversity was just shuffling the sequences that are available at this point. In addition, you have the so-called junctional diversity. Which is 10 to the 7 times more. or several times 10 to the 7 times. So 10 million to 30 million times more, not extra 30 million, but this many times more. Okay, so altogether you could generate a 10 to the 13 or even more different diversities. Now, this I call the diversity which probably is enough to generate an antibody to any type of molecule that you can picture in your mind or synthesize here on Earth or in the solar system. Okay, so how is this going to work? First, I would like to show you the mechanism of genomic recombination. We will not stress on this too much, but it's interesting. So quickly, there are recombination signal sequences at the protein encoding gene segments, okay? And these are set up so that there is a heptamer close to the coding sequence here and there, and then there is a spacer of various sizes. On one side of the rearranging sequence, it's 23 base pair long. You know, the noise is getting too high. So 12 base pair on the other end. And if this is the rearrangement and a 23 base pair spacer in between the nonomer and heptamer sequences are close to 12-mer spacer, again in between the nonomer and the heptamer sequences, these two gene segments could be joined. So V lambda segment could be joined with the J lambda segment, and this is called the 12-23 rule. So these guys could be joined, okay? Now what about the J segment of the heavy chain with the V segment of the heavy chain? Both have a 23 base pair spacer in between the 9 mers and 7 mers. These guys are not going to join together, but the D segments have a 12 mer on both sides. So this guy, J segments can be joined with the D segment when it's paired with the J segment, it joins the V segment because the 12-23 rule applies. This is not very easy to understand from the text, so that's why I want you to understand here and now. So the rearrangement is done by an enzyme complex. When you bring together these segments, what you will get is the V segment and the J segment put close to each other, and the loop out sequence, which contains various number of different segments in between. Sorry, my drawing. Separated by these repetitive type stem loop sequences and so on. So the good type of sequence is when you have a short spacer, like a 12-mer, in combination with a 20-mer, 23-mer, and then you have this sequence of DNA, which could be quite long. Now this is a different sequence from a stem loop structure, which is of equal size, okay? And the enzyme complex will prefer the non-symmetrical 12-23 base pair stems with the loop structure over this. Alright, so if you got it, fine. If you don't, you still could be very good dentists. But you read it, and if you want, you understand it. Now, so this rearrangement is allowed, this rearrangement is not allowed, because this is going to generate a stem loop with an equal length stem. These guys generate stem loops with a different size stem. Jesus Christ, there's no... What's that? This figure is nicer on your computer. This just shows the same thing, but that's really bad. Okay, don't worry about this part.
We should ignore that?
I want to explain to you the source of junctional diversity, okay? Forget everything about rearrangement.
Yeah.
I want you to concentrate on this part of the figure. When the rearrangement started, the recombinase enzymes will cut... we'll cut the DNA, and double-stranded breaks are gonna form, and the DNA attacks its own strand by a nucleophilic attack, and you will generate a hairpin structure. You know hairpins. Ladies know hairpins. Gentlemen know hairpins also, but from a different experience. We see them as obstacles. We may see them as opportunities or possibilities to look beautiful. So this is really small, so I try to reproduce this. So here's one fragment. I don't want to hear laughs because they look okay, except that, of course, everybody has a different opinion of these. So this is called a hairpin, and this is segment 1, and this is number 2. Okay. So this is what is generated by the recombinase, and then after this there is a protein called Artemis, which is, you don't have to remember the name, which randomly cuts up this hairpin structure, which is, it can cut here, here, or even further down here. And what happens to this DNA? Let's say it cuts here, far away, okay? So I'm trying to draw the situation what happens if it's cut here. You have this 3 nucleotide sequence here, and then a long unpaired DNA, and it folds over. So 1, 2, 3, 4, 5, 6, 7 times 2. So a long non-paired DNA. But it's random. You could open it up here and have a short sequence only. This sequence was never present before in the germline. Also, if you think about it, these guys are palindromic sequences, so that they are the same from both sides read, because they were originally folded like this. So they actually can pair with each other. Same thing here. Now, this sequence is filled in by nucleases, by polymerases, okay? So this is a part of DNA that was never there before in the germline, newly generated and right at the junction sites. And these are the P nucleotides that were not there. Let's see how it looks here. So the same thing is shown here. It's shown here. It's got this AG is folded up here, TCGA, and these P nucleotides were not there. This was the first step is rearrangement, second is opening up the hairpin, and the third step is to add more nucleotides. There is an enzyme called TDT, which is a polymerase, terminal deoxynucleotide transferase, which adds non-templated nucleotides in a random fashion. These are the last step of the junctional diversity process. So the bottom line is using enzyme systems that will randomly open up these hairpins, and the TDT enzyme that randomly adds extra nucleotides, which are called non-templated or N nucleotides at the junction site, will generate extra sequence never there before, and added by an entirely random fashion. Therefore, there are no rules for them. There's a lot of diversity depending on how long these all are. So, if we get that, we can go on and look how the cells develop. This is somewhat more of immunology here. We'll keep this very simple. There are 2 big phases of lymphocyte development. One is in the bone marrow, and the other is going to take place in the secondary lymphoid organs. The first one definitely needs no antigen exposure. This is an inbuilt process. It's generated automatically. It's programmed no activation signals from either self or foreign antigens are required. And then in phase 3, the lymphocytes that developed using this gene rearrangement process, they develop the cell surface receptors. They will leave the bone marrow and go to the spleen and other lymphoid organs, and these B cells can get survival signals. If they manage to mature at this stage in phase 3, when they leave the bone marrow, they're not mature yet. Okay? In phase 4, they get the maturation signals, and in phase 5 and phase 6, they will look for infective agents in the periphery, and in phase 6 they get activated, they go to form germinal centers, and they become baby plasma cells at the end stage. Okay. The stages of B cell development could be followed by the gene rearrangement process. There are 3 different cell types that I want you to remember. One is the pro-B cell, which have practically no non-rearranged gene segments, or the DJ rearrangement starts. The pre-B cell, which has already rearranged the heavy chain, and the immature B cell, which rearranged both the heavy and the light chain. There are certain cell surface molecules and IL-7 receptor interaction that is required for the survival of the developing lymphocytes. Now, obviously, these gene rearrangement processes are difficult to make. Since these are random procedures, you can lose nucleotides, you can gain nucleotides within the coding sequence. So the open reading frame, the coding frame can be messed up very easily. A lot of times, you join the gene segments and you're not going to get any good sequence out of it. That's what makes the heavy chain procedure slightly inefficient. Okay? If on one chromosome DJ and VDJ rearrangement occurred and you don't have a viable transcript that could be translated into a normal, good, okay, full-length protein, then the B cell dies. If this happens first time, the second chromosome still can generate a viable rearrangement, and then the B cell is going to survive. If both events on chromosome 1 and chromosome 2 produces non-viable proteins, non-translatable sequences, then the B cell dies. About 50% of the B cells is going to die. But how does the B cell know whether the protein is good or bad? The protein is tested by its ability to bind a protein chain, which looks like the light chain. If the protein is translated correctly, it will fold up in a sequence or in a structure which will be able to bind to the light chain. Now, the light chain is not available yet, therefore the B cell produces a light chain-like molecule called the surrogate light chain. This is non-polymorphic, and this is going to be forming a complex with the just newly made heavy chain, and associate with the signaling subunit of the BCR, which is called IG alpha and IG beta. Any of these chains are missing in a person by mutation, the surrogate light chain components, IG alpha or IG beta, the person is not going to have viable B cells in their blood or in their lymphatic organs. If, however, this procedure is successful, then it will shut off further rearrangement of the heavy chain in a process called allelic exclusion. So what is allelic exclusion? That's horrible. Let's open the windows there, you guys, or something. Sorry. Somebody is really out there. It's only 30 minutes. You guys need to be stronger, tougher, and more interested. So if the so-called pre-B cell receptor is able to form in between the surrogate light chain and the newly formed heavy chain, then some signaling takes place which will shut off further rearrangement of the heavy chain on both chromosomes, which is called allelic exclusion. It gives survival signals. It will induce the cell to proliferate, and it will start the rearrangement of the light chain. After this, the light chain is rearranged and forms a viable kappa or lambda chain. It is somewhat better situation because there are no D segments here, so as long as you have a free kappa chain or a free J segment, free variable and a free joining segment, the B cell can try and form a good protein. So therefore, approximately 100% of the light chain rearrangements will bring a bona fide protein. So the BCR, the B cell receptor, can form. First, the kappa chain is rearranged on one chromosome. If that's not successful, then the kappa chain on the second chromosome is rearranged. If that's not possible, then the lambda chain starts rearranging, and if that fails, on one chromosome you can still get a second chance of the lambda chain rearrangement, and only after the cell dies by apoptosis. So once the BCR is assembled, the B cell is ready to leave the bone marrow. Two checkpoints, one at the pre-BCR stage, then the cell proliferates and starts rearranging the light chain, and the second checkpoint is whether or not the B cell receptor is able to form a complex with the Ig alpha-beta heterodimer and give survival signals to the developing B cell.
So different checkpoints than the cell proliferation checkpoints. So
Yes.
cell cycle has
Here you have a large proliferation procedure. From a single cell you can get approximately 100 daughter cells, and in this 100 daughter cell the light chain starts rearranging. So for sure, out of the 100 cells, you will have quite a number of cells that successfully rearrange the second one. This is like you buy a cell line from a company, and then you extend those cells and make 20 vials for yourself to save it for further playing with the cell line, so that you don't lose it. The same thing happens here. You amplify one successfully rearranged light chain containing cell type, so that you can mess up here the light chain rearrangement and still have a few cells that survive.
So the V3B is the variable 3B cell, not the first one.
Which one?
This
Oh,
is-
the V3. This is a protein which is called the variable pre-B cell receptor protein. It looks like the variable segment of the light chain. Okay? Instead of having a constant region and the variable region for the light chain, these are two different proteins. They associate, and together they join the heavy chain to form a light chain-like structure. Okay, biochemistry is not more or less over. The next step is for the B cell to get survival signals after they leave the bone marrow. It's very important to remember that when the B cell leaves the bone marrow, it's still functionally inactive. It needs to receive survival signals, and it receives that from the follicular dendritic cells present in the spleen and some other lymphoid organs, secondary lymphoid organs. And these B cells follow the same chemokine gradients that we already discussed. CCL13 is actually the major type of chemokine expressed by the follicular dendritic cells that will drive the B cells into the B cell area of the secondary lymphoid organs. CCL21 and 19 are produced by the dendritic cells in the T cell area. These are also going to draw B cells out of the circulation. So they step out to the halves and then they go to the follicular dendritic cells. These immature B cells then only live for a few days. Okay, so 4 or 5 days is the lifespan of the immature B cells. After they got the survival signals, they will live up to 2 months even. So they will have plenty of times to look for infective agents. Okay, so the mature naive B cells versus the immature have a large difference in life expectancy. And only those B cells survive that will get survival signals basically in the form of a cytokine called B cell activating factor or BAF. This is a TNF receptor family member cytokine. And they then become mature B cells, naive mature B cells. They express IgM type and IgG type cell surface B cell receptors. How is it possible to actually express two types of different receptors with two different constant region and heavy chain? The answer is here. When the primary transcript is generated, you could have an alternative splicing and polyadenylation, and if the polyadenylation sequence after the new type exons is used, then you get an IgM type BCR on the surface. If you get the polyadenylation sequence used here, and the splicing occurs between here and the donor site here on the delta chain, then you will get a delta mRNA on the cell surface. And they are co-expressed in the mature naive B cells. At this stage, when the B cell leaves the bone marrow, it's called a transient B cell, and after that, it is called the mature naive B cell, and they got the survival signals. Okay, so we are more or less done with the B cells. Now let's see about the T lymphocytes.
Oh, wow.
It's very exciting.
It's genetics.
What is more important, the B cells or the T cells for the survival of a person?
50-50.
B cells.
Which one?
T cells.
Okay, who votes for the B lymphocytes being more important? Who thinks that? 1, 2, and I'm against. I think the T cells are more important. Why? If you don't have T cells, you will definitely die, because both the T cell responses are effective, and also the B cell responses, because most of the B2 type protein-induced B cell responses are dependent on the presence of T cells. So the lack of T cells will generate a syndrome called severe combined immunodeficiency syndrome, which is fatal, basically. So you cannot live without these T lymphocytes. This is a well-known picture of the thymus. This is the cortical region. Here you have thymocytes. The medullary region has this basal corpuscles, dendritic cells, the medullary epithelial cells, while the cortical region has the epithelial cells, which are called the cortical epithelial cells. When we discuss later on the positive and negative selection processes, we will need to know these cell types. That's why I'm showing that. But basically, I don't want to go through it again and again very much in detail, but the point is that the alpha and the beta chain of the TCR is generated in a very similar process to that of the heavy and the light chain. Also, the alpha chain is a homologue of the light chain, and the beta chain is a homologue for the immune globulin heavy chain. And how we know this is because the alpha chain has only J and V segments, like the light chain, and the beta chain has these diversity segments, not too many, only 2, but nevertheless, they are built the same way, like the heavy chain. There is a cartoon or a movie which I will upload eventually to the dentistry folder if it's not there already, maybe it's there, which shows that the non-committed pre-T cells or not even committed lymphocytes that are going to be taking the path of T cell development enter through the venules in the thymus. They go towards the cortical areas, they meet with the cortical epithelial cells, they rearrange the TCR genes, they have a checkpoint just like the B cells, the pre-B cells, they have a pre-T cell checkpoint in which the beta chain integrity is checked, and then they start rearranging the alpha chain. The secondary checkpoint is designed to check whether the alpha and the beta chain forms a viable heterodimer, and if so, then the T cell is ready. When the T cell leaves the thymus, it's already a mature T cell. But before we go too far, I want to show that the pre-TCR was built from these B, pre-B, lambda 5 segments. The pre-T cell receptor is built from the so-called PT alpha, which looks like a part of the alpha chain. It's also not polymorphic. Its function is to recruit the CD3 signaling complex. When this complex would assemble, and then you get signaling which will inhibit the beta chain rearrangement, it will induce the proliferation and survival of the T cell. 10 minutes left only, okay? Go to your telephones and get lost. What can I say? Don't disturb the 3 people who is listening, okay? I love students, especially, okay. So the function of the pre-TCR is the same as the function of the pre-PCR. That is, it switches off further rearrangement of the already rearranged chains. But there is some differences. This signal starts the expression of CD4 and CD8, the well-known co-receptors of T cells. At this stage, in the pre-B cell stage, there's no CD8 or CD4 expressed on the B cells. When this receptor assembles, you get both expressed at the same time, okay? So the double negative CD4, CD8 negative cell becomes double positive. Here is the procedure, okay? Let's see what happens. And then, after that, at the second checkpoint, you will have the alpha chain, the beta chain assembled with the CD3 complex, gamma, epsilon, and zeta, as we learned from other lectures or seminars. Now, how do we know that the T cell going out from the thymus is going to be able to recognize the MHC molecules in combination with bacterial or viral derived peptides? First off, the T cell that is going to function in the periphery needs to remember the self-MHC. So there is a procedure called positive selection to make sure that the T cell has a certain amount of reactivity with the self-MHC. This is very important from the point of bone marrow transplantation. This is why you need to have relative, or you need to have an exactly matching random bone marrow donor, because the antigen presenting cells of the recipient needs to be able to talk to the T cells of the donor. So this is really an important thing. Only those T cells will survive this step of T cell development that will have a certain amount of affinity to the self-MHC, self-peptide complex, and this is tested by the cortical epithelial cells. Again, this procedure is called the positive selection. And this is the molecular basis for the so-called MHC restriction, okay? So this is what ensures that out in the periphery, you will have all the T cells basically recognizing your own MHC, and this is going to be the basis that in the periphery you will have self-MHC polypeptide complex is recognized by the T cells. Otherwise, it's not.
So, sir, the MHC should be completely matching or?
For bone marrow transplantation, in the old days they did that. If they don't have a matching donor, then they might go for less. Okay, 5 match out of the 6 is better, but these days they can even do haploidentical donors, which means that one set of MHC is matching, the other is different. But as more and more mismatch you have, the more and more chances are there that you will have GVH, graft-versus-host disease. But at the end of the story, we probably are going to talk about bone marrow transplantation as well, briefly. So, positive selection, the selection of all the lymphocytes, the developing T cells that will have some affinity to MHC, self-MHC. Those lymphocytes that will not have any affinity to self-MHC, they will die. Only about 1 to 2% of any of the developing T cells will react with any particular MHC, but we will have approximately 10 different MHCs on the surface of our cells. So MHC2 at least 6 different types, MHC1 another 6. So maybe 10% of the cells are going to survive this. So this was MHC restriction is one step important, and the positive selection also ensures that the double positive CD4, CD8 positive T cells are going to become single positives. That's really almost the last slide, so just bear with me for a few more minutes. If the double positive T cell recognizes an MHC-bound peptide on the thymic epithelial cell, then the CD8 is going to interact with the alpha chain of the MHC class 1 molecule, and this interaction gets fixed, and the CD4 expression gets downregulated. And the cells that come out of this interaction are going to be single positive CD8 cells. However, when and if you get a TCR which finds a peptide, MHC complex for which they have a relatively reasonable affinity, then the CD4 molecule at the same time can interact with MHC2. Domain here gets fixed and CD8 is downregulated, and you get a single positive CD8 positive helper type T cell coming out of the thymus. How do we know that? Because there is a syndrome called the Bear lymphocyte syndrome, in which MHC type 1 or type 2 is not expressed. If the person lacks MHC class type 1 molecules, then CD8 T lymphocytes are very seldom in their bodies, meaning that MHC peptide complexes are not seen, CD8 doesn't get fixed. However, MHC class 2 type peptides are there in this person, only MHC class 1 is missing, so CD8 cells can form. Okay? So these people will have a predominance of CD4 cells. If you don't have MHC class 2 in the person, but you do have MHC class 1, the opposite. You will have no CD4 T cells, but you will have CD8 T cells. And if you don't have T cells at all, what you get is a bubble boy, severe combined immune deficiency boy, which results in an extreme sensitivity to all sorts of infections. And these are the horrible figures of different types of SCT people. This is the little boy who lived for years in a sterile tent, basically. Sterile air is blown in here. So from his birth, he spent not a single minute out in the real environment. Once he was let out to play with his sister, where NASA actually designed a scaphandre for him, and they could run around for a few minutes. Otherwise, he only could interact with people through this plastic bag. Okay? Tough life. The hallmark of SCT is basically this thrush, which you can see in the mouth. If you see this in a kid, it's very likely that cellular immunity is severely hampered. The neutropenia or SCT phenotype will come with this. This is a fungi, pneumocystis infection, and all sorts of diseases that you would see in an AIDS patient can develop in these people because they don't have T cells, just like AIDS patients. They don't have CD4 positive T cells. When the RAG enzyme that does the recombination is defective, you get kids with the so-called Coman syndrome, and the skin becomes really reddish, like in a GVH patient. Some autoreactive T cells still get generated in the absence of the RAG enzymes, and the skin really becomes reddish.
Before there, to understand that this
They knew these patients, but they died very quickly, okay? So that's the situation.